This is Elizabeth Montgomery, master's student in the Land Resources and Environmental Studies program at Montana State University. This presentation is for biodiversity samplings and methods. My study is a comparison of the benthic macroinvertebrate community above and below the Hyalite Creek Dam. So why do we want to compare the benthic macroinvertebrate community above and below Hyalite Creek Dam? Well, we know that dams can alter streams physically, chemically, and biologically, and that they could potentially degrade this benthic macroinvertebrate community and their habitat downstream, which would affect riparian productivity and also food availability for native fish and other aquatic predators. An example of the kinds of effects dams can have on the landscape. Here's a large dam on the Euphrates River in Syria, and you can see the physical changes even from space. So you can only imagine what this does to water quality and chemistry and habitat on a smaller scale. And in case you are unfamiliar with the benthic macroinvertebrate community, they are some of the primary consumers of freshwater environments. Here are some of our more common orders: Plecoptera stonefly larva, mayfly ephemeroptera larva, chironomid midgefly larva, Trichoptera caddisfly larva. These animals spend the early part of their life in the freshwater environment before metamorphosizing into flying adults. One really important aspect of benthic macroinvertebrates is their capacity to be used as bioindicators or bioassessment of stream health. And the BMI community has been used as indicators of stream health since the early 20th century. Now, we know that dams can affect water quality by increasing or decreasing water temperature, depending on if your outflow is at the top. Surface waters would be warmer, or if it's from the bottom, the temperature can actually be cooler. Dams can affect dissolved oxygen concentrations. They can alter flow patterns depending on when releases occur. They, of course, prevent transport of sediment and other materials downstream. We all know that dams over time become sedimented and sometimes need to be dredged out. There's also evidence they reduce leaf litter breakdown rates, which affects nutrient cycling and productivity of our streams. All of these impacts, and probably others, can have cumulative effects on then the biological communities, including the benthic macroinvertebrate diversity downstream of dams. So, are we seeing any of these effects in Hyalite Creek below the Hyalite Dam and Reservoir? It's the perfect study site to investigate these claims. So, the creek and reservoir are in the Gallatin National Forest. It's a montane stream at 6,700 feet in elevation. And the dam was first constructed in the 1940s and since enlarged in 1993. And here you can see a photo we took of the dam on the ground. This is at the downstream extent of the reservoir. And then the figure on the right shows a Google Earth image of the reservoir, and if you look closely you can see our upstream sites at the bottom end of the image and our downstream sites at the top end of the image. How are we going to go about in achieving this goal? We're going to apply statistical analyses to compare BMI abundance, species richness, and diversity, as well as community composition between the up and downstream sites. We will also use analyses to compare water quality conditions up and downstream of the Hyalite Reservoir, including water temperature, conductivity, and flow. From both 115-meter transects, we collected 4 samples across a distance of 10 meters, and each sample was 25 meters apart from each other. This is a modified version of the EPA's protocol for bioassessment in wadable streams. I'm going to begin the results with our water quality data first before moving on to our benthic macroinvertebrate community data, just to get a sense if the dam is possibly having an effect on these water quality conditions. And as we can see, water temperature was significantly different upstream than downstream, being much cooler at the upstream site than the warmer downstream site. And this was about a 4 degrees Celsius difference. Conductivity was also significantly different between upstream and downstream. Conductivity was much higher downstream of the dam and lower upstream of the dam. Stream flow was also significantly different between sites, being generally higher downstream and lower upstream. And I do want to point out that we did observe water actively flowing from the dam. Now on to our biological results, which is the benthic macroinvertebrate community. We did an ANOVA chi-squared test and observed that BMI abundance was significantly lower at the downstream site than the upstream site. BMI richness, which here was the number of distinct orders, was not significantly different. I then broke down BMI abundance by order, which we have across the top: Plecoptera, Ephemeroptera, Trichoptera, Diptera, Coleoptera, Acari, Tubularia, Copepod, and Amphipods, to see what percent of the variation in the abundance of each of these orders could be explained by our different treatment variables. And what I did find was that some orders seem to be more affected by treatment type than others. Plecoptera and Trichoptera, for example, were significantly more abundant upstream than downstream. And Acari and Copepod were significantly more abundant downstream than upstream. I will point out the difference of scale between these orders. For Plecoptera and Trichoptera, 10 to 30 were found downstream, where 30 to 50 were found upstream. And for Acari and Copepod, our total numbers at both sites were under 4. So the difference between upstream and downstream could just as well be detection errors, particularly as these were far more rare across the environment. An attempt was made to also parse out what percent of variation could be explained by the different water quality conditions, but what I found was that they aligned very closely to the percent variation explained by the treatment, which is upstream or downstream. And this indicates that there's interference because temperature differences align with the treatment variable, so it's difficult to parse out which one has the strongest effect, and a multiple regression analysis would probably be better to help explain some of this variation. Interestingly, across the board, temperature and conductivity did seem to explain a lot more variation than stream flow, which only had a significant correlation with 2 orders, whereas temperature and conductivity had significant correlations with 4 different orders. Did species diversity differ between the upstream and downstream site? The answer is yes. Both Simpson's diversity index and Shannon's diversity index showed that the downstream site had significantly lower species diversity than the upstream site. I applied a Curtis-Bray dissimilarity analysis to compare the alpha versus beta diversity. Were differences in community composition between the upstream and downstream site greater than differences among the 4 samples within the downstream and upstream sites? And while we can see that dissimilarity was highest between upstream and downstream, 0.32, this did not prove to be significant, So in the discussion, I will go over how a number of our dependent variables varied significantly between the upstream and downstream site. The downstream site had significantly higher temperatures, higher flows, and higher conductivity, indicating significant changes to the freshwater habitat below Hyalite Dam and reservoir. It also appears there is a biological response to this changed habitat, wherein downstream of the reservoir we found lower benthic macroinvertebrate abundance, lower species diversity, and fewer numbers of a few specific orders such as Plecoptera stoneflies and Trichoptera caddisflies. This evidence leads to the conclusion that it is highly likely Hyalite Dam and reservoir affect the BMI community of Hyalite Creek, especially within at least 300 meters downstream where we were surveying. This is due to our observed significant differences in water quality conditions up and downstream, as well as significant differences in BMI abundance, species diversity, and number of Plecoptera and Trichoptera between sites. An insignificant results in our Curtis-Bray dissimilarity index indicated that there could be a high level of habitat patchiness wherein our 4 samples from a given site had variance that was not less than the variance between upstream and downstream. Another consideration is what is the spatial extent of the effect. If we do determine that the Hyalite Creek Dam and reservoir affect the benthic macroinvertebrate community, it would be really interesting to further investigate how far downstream these effects are observed. And finally, in this study, we only went to order due to time considerations in the field. However, if we performed a higher taxonomic effort, identifying each individual to family or genus, it would be very interesting to see how this would affect our results. If we still saw species diversity differences between up and downstream, this would also allow us to further investigate functional feeding groups and also tolerance values, which are not really applicable at the level of order. Another reason the higher taxonomic effort would be interesting is you could carry out a study to determine what degree of taxonomic effort is required to first notice disturbance. So, for example, whereas a community group might be able to go out and carry out a study very similar to ours, identifying everybody to order, can they effectively identify disturbance to the community, or is a higher level of expertise and identification necessary to measuring these kinds of impacts? That sums up this presentation on comparing the benthic macroinvertebrate community upstream and downstream the Hyalite Dam. I hope you learned something, and if you want to learn more about this topic, you can look forward to my complete report.